Dear children, this is a future-ready science lesson for school children. This is about recombinant DNA versus genetic engineering for high school students. If you understand these things, you will be ready to do research when you go to higher classes. I am C.G. Nagaraj. Based upon my experience of 60 years of teaching, I am producing this particular video explaining what is recombinant DNA and what is genetic engineering. You need not know much details, but you should know what these ideas in science are. There is a single-celled organism called E. coli which lives in our intestine and in some other animals. It's a tiny cell, but it makes the big signs. When I say big signs, over the world, people are spending millions of dollars to try this organism to do many things. This tiny cell shows how even small life can create huge progress. Why E. coli is used in research? It is used in recombinant DNA, used in genetic engineering. These two together become biotechnology. What do you mean by recombinant DNA? We can add new genes to E. coli. Then E. coli will start producing like a factory. These are very, very fascinating areas. You should try to know what these areas are. Let us try to know what is recombinant DNA. The new DNA created by joining genes from different organisms. That is recombinant. That means recombination of DNA molecule. In the living beings, there is what's called DNA, and that is deoxyribonucleic acid that makes the chromosomes. Chromosomes may contain thousands and thousands and thousands and thousands of genes. Each gene may contain many chemical molecules. They are all non-living things. So living things have inside them non-living DNA. This particular DNA carries information for the entire life processes. Then what is genetic engineering? This process uses many techniques like an engineer uses technology. Technology is used to cut, combine, join DNA molecule to produce useful products. In one sentence, if I want to tell the difference, recombinant DNA is the product formed by joining DNA from different organisms, while genetic engineering is the multi-step process of manipulating genes to create recombinant DNA and produce new traits. Let me again explain to you what is recombinant DNA. A new DNA formed by combining genes from two different organisms. Organism means I am talking about living beings. Scientists cut a gene from one organism and paste it into the DNA of another organism, and this becomes a recombinant DNA. If you take a daily example, take a page from your friend's notebook and paste it into your notebook. Then your new notebook will have new information. That is a simple example. In scientific example, if I want to give you, human insulin gene is made to recombine with the gene of E. coli. Then G. coli will produce insulin. Let me again explain to you what is genetic engineering. The complete process of changing the genes of an organism that includes selecting a gene, cutting it or splicing it, joining or combining it, inserting it, and make it to express. If I take an example from your own school level, like you plan, cut pictures, paste them, and decorate that into a scrapbook. It becomes a new book. See, papers, pictures, all these things put together, it becomes a new one. It's a technique. That means you cut, you paste, you select, you plan. All these things include engineering work. Example, new traits like pest resistant. That means in the plants, many insects may eat them. To resist it, they produce a new trait in the plants. Insulin is produced by bacteria, E. coli, and the golden rice is made. Today you find in the shops different varieties of rices, which are all made by means of genetic engineering and recombinant. Let me give a summary of what I told you just now. Recombinant DNA is equal to the new mixed DNA. Genetic engineering is equal to the technology used to make that new DNA. Both are powerful tools in modern biology, help humans to produce medicines, better crops, and solve diseases. Science is not magic. It is understanding and modifying life at the gene level is genetic engineering. This has a special reference to E. coli and biotechnology. Biotechnology is a multidisciplinary field that involves the integration of E. coli genetic material and engineering sciences in order to achieve the application of organism, its parts, for products and services. Specialists in this field are known as biotechnologists. You can plan to become a biotechnologist if you understand what is recombinant DNA, what is E. coli DNA, how to cut it, how to paste it. At least if you know the information before you go to college, you can become a biotechnologist. Here, I will ask you one simple question. Did our ancestors knew genus to genus engineering? Genus to genus engineering. Matsya, Kurma, Varaha, Narasimha, these are very mythological gods we approach to worship. We call them in Dashavatara. Did these images are real or made or really our ancestors knew about recombinant DNA, genetic engineering and technology to make genus to genus engineering, that's a big question mark. You have all the way